Assalamualaikum and hi. Today I will present about my mini project, which is under the code ECG 263 Soil Engineering Laboratory. First of all, my name is Muhammad Zafri bin Zahrul Nizam. My student matrix is 2021816242 and my group class is J4CAC 1104B2. In this laboratory activity, students will study about the effectiveness of additive substance to the soil. This mini project consists of a laboratory activity of particle density test and Casagrande test. The substance POBA will be used in this laboratory activity with the percentage of the additive substance will be considered. The objective uh, of this laboratory activity is to determine the particle density and specific gravity of soil with an additive waste material POBA. And second is to determine the plastic and liquid limit of a soil sample with an additive waste material POBA. Background of study. In this laboratory project, the POBA has been chosen as my additive waste material to improve soil stability. 7% of this substance is used in the soil and then will be compared with the soil without any additive substance. Next is the apparatus. The apparatus of particle density. First, density bottle with stopper having capillary hole at its center. Wash bottle. De-add distilled water, alcohol, ether balance, vacuum sets, constant temperature water bath, specimen, container, weighing balance, and oven. For the procedure, first, uh, students need to prepare the apparatus. First, clean and dry the density bottle and stopper properly. Weigh the dried bottle with stopper and record the mass M1. Take about 10 to 20 grams of oven-dried soil sample cooled in the desiccator. Pour it carefully into the density bottle. Weight the bottle with soil and stopper. Record the mass M2. Pour about 10 milliliter de-add distilled water in the bottle to soak the soil. Leave it for a period of 2 to 10 hours. Then, pour some more distilled water to make the bottle half full. Remove the entrapped air by applying partial vacuum. Pour some more distilled water into the bottle and fill it completely without any entrapped bubble. Put the stopper on. Keep the bottle on the stand in constant temperature water bath for 1 hour. Take out the bottle from water bath. Wipe it clean and dry it from outside. If the capillary of the stopper is not full, fill it with drop of distilled water. Make again. Make sure the bottle and stopper are clean dry. Weight the bottle filled with water and soil sample with stopper. Record the mass M3. Empty the bottle and clean it properly. Fill the bottle entirely with distilled water. Make sure there are no entrapped air bubbles or otherwise the partial vacuum has to be used. Put on the stopper and wipe the bottle dry from outside. Now, wait and record the mass M4. Again, empty the bottle and dry it up properly. Lastly, repeat the step 2 to 11 for 2 observations to obtain an average specific gravity of the sample. So, here is the result of the particle density test. Based on the discussion, the moisture content of soil, also referred to as water content, is an indicator of the amount of water present in soil. Students need to find moisture content for allowing irrigation to be quantified in advance. The, the specific gravity is determined by the calculation. Specific gravity is important to know because it helps a lot of engineers understand how porous the soil is. So, the conclusion is students need to able to conduct the laboratory activity and able to get the data properly, and the objective of the laboratory is achieved successfully. In the next activity is the apparatus of Casagrande test. The apparatus is Casagrande liquid limit device, grooving tool, spatula and palette knife, moisture can, wash bottle containing distilled water, weighing balance, glass plate, and drying oven. Next, for the Casagrande procedure. First, students need to prepare the apparatus. Take 200 grams of the soil and place it onto glass plate, pass non-40 sieve. Thoroughly mix the soil with a small amount of distilled water until it appears as a smooth uniform paste. Cover the mixture with cellophane to prevent moisture from escaping. Label and weight 3 empty moisture can. Adjust the liquid limit apparatus by checking the height of drop of the cup. The point on the cup that comes in contact with the vase should rise to a height of 10 mm. The block on the end of grooving tool is 10 to 13 mm height and should be used as a gauge. Practice using the cup and determine the correct rate to rotate the crank so that the cup drops approximately 2 times per second. Place a proportion and previously mixed soil into the cup of the liquid limit apparatus at the point where the cup rests on the base. Squeeze the soil down to eliminate air pocket and spread down into a cup of depth of about 10 mm as its deepest point. The soil pad should form an approximately horizontal surface. Use the grooving tool carefully. Cut a clean straight groove down the center of the cup. The tool should remain perpendicular to the surface of the cup as groove is being made. Use extreme care to prevent sliding the soil relative to the surface of the cup. Make sure that the base of the apparatus below the cup and the underside of the cup is clean of soil. Turn the crank of the apparatus at a rate of approximately 2 drops per second and count the number of drops n it takes to make the two halves of the soil pad come into contact at the bottom of the groove along a distance of 13 mm, half inch. If the number drop exceed 50, then go directly to step 8 and do not record the number of drop. Otherwise, record the number of drop on the data sheet. Take a sample using the spatula from edge to edge of the soil pad. The sample should include the soil on both sides of where the groove came into contact. Place the soil into a moisture content. Immediately wake the moisture can contain the soil. Record its mass and place the can into the oven. Leave the moisture can in the oven for at least 16 hours. Place the soil remaining in the cup onto the glass plate. Clean and dry the cup on the apparatus and the grooving tool. Remix the entire soil specimen on the glass plate. Add a small amount of distilled water to increase the water content so that the number of drops required to close the groove decrease. Repeat step 6, 7, and 8 for at least 2 additional trials producing successively lower number of drops to close the groove. One of the trials shall be for a closure requiring 30 to 40 drops, one for closure between 20 and 29, and one trial for a closure requiring 10 to 19 drops. Determine the water content from each trial by using the same method used in the first laboratory. Remember to use the same balance for all weighing. So here, this is the result of Casagrande test. So for the conclusion, the objective of the laboratory is achieved successfully, and there is also safety precaution is need to be considered.